Dr. Roberto Amato, who's a scientist at, who's the lead of the analysis group at the Wellcome Sanger Institute in the UK, and he was leading a number of research studies on the genetic markers of piperacin resistance, which we conducted largely in collaboration with studies in Cambodia as well as other neighboring countries. So, Roberto, would you like to take over?
Thank you. Can you see my screen and can you hear me? Can you see me?
Yes.
The usual.
Yeah.
Great. Okay. Thanks, Professor Olivo. Thanks for the introduction. It's really a pleasure to be here. I'm realizing what I'm going to present today is very complementary to what we heard so far. We heard the view from our perspective, from the human perspective, what's happening in the field, the number of cases, number of deaths, and great to hear the numbers are going down. What I'm going to present is kind of complementary to that. We're going to see from the parasite point of view, as in from the parasite genomic perspective, molecular perspective, what was happening during those the past 10 years or so. By the way, welcome to my bedroom. I realize I should have picked a better background than that. So because I'm going to cover a different aspect, I thought I'll start from the, from first principles, really. I'm going to start from the basis. I don't think Kelch needs an introduction as such, but just in case, Kelch13 is a gene in the parasite genome that causes resistance to artemisinin. We actually know that it's a causal relationship. It's not just an association. This paper came out in 2013 from the Institut Pasteur. And shortly after, if my memory is correct, which never is, no more than a couple of months later, a few months later, this clinical study led by Liz Ashley and Morrow, I'm showing a figure from the paper here, confirmed that in the field. And two observations came out of that study. First, wasn't just a mutation in Kelch13. So we're kind of going away from this idea of, CRT, for example, where K76 or 76T cause resistance to artemisinin. The picture seems to be a bit more complex. So there was difference number one. Different number two was that at the time, sort of surprising, or maybe not, this mutation seems to be arising independently, as in we're not really looking at the spread of this mutation. The spread from a molecular perspective wasn't really spreading, but was emerging in large part of Southeast Asia, in particular around the Greater Mekong Subregion. Again, this figure, I think you're familiar with that, but just to cover the bases, as I was saying, shows the parasite clearance half-life that's measured in the patient in the clinical study led by Liz. Numbers above 5 hours that indicate parasites that are resistant to what we call resistance to artemisinin. Below 5 hours are parasites that are sensitive to artemisinin. One reason for putting this figure up is to remind ourselves that even if we do know that this is a causal relationship, as in you can edit the genome of the parasite, introduce this mutation, and you will observe the phenotype, this is never clear-cut. Look at this parasite here, for example, despite having no mutations, no Kelch mutations, still have long half-life, as well as this one. This is our well-known 580Y. With short half-life. I'm not saying anything new there, but just keep in mind that genetic molecular markers are great proxies, but never shouldn't be taken as the absolute truth. So that covers Kelch and artemisinin resistance. The other part of the story is around piperacin, plasmepsin and piperacin resistance. In 2017, we published this study, again, another clinical study, where we observed that this amplification of two genes in the parasite genome called plasmapsin 2 and plasmapsin 3, they sit next to each other on one of the parasite chromosomes. When you have tandem duplication, that is, instead of having one copy of each gene, you have multiple copies, 2 or more of those genes, not only you observe a piperacin IC50 going up, that is in vitro, measured in vitro and ex vivo, essentially is how much drug you need to kill the parasite. So this number is going up. This on the left are parasites with a single copy of those genes. On the right are parasites with multiple copies of the genes. And as you can see, the phenotype goes up. But more importantly, parasite with multiple copies of the genome seems to be causing complete treatment failure, as in the chance of the patient getting well after being given artemisinin piperacin, which was the frontline treatment at the time, as we just heard from Dr. C, goes down by more than 6%, in fact. So it's almost a 50-50 chance, even worse than 50-50 chance, whether you'll be cured with DHA PPQ if you're unlucky to carry a parasite that's got both Calc13 mutation and plasmapsin amplification. I don't think I've said anything new, but I think that was important to introduce this plot, which is analogous to other plots we've seen during the day since this morning. So this is the frequency of artemisinin and piperacin resistance in Western Cambodia between 2007 and 2013. As I was saying, but this is looking from the molecular perspective, as in looking at the parasite genome and see what the frequency of this mutation, again, Calc13 mutation for resistance to artemisinin and plasmapsin 2 and 3 amplification for resistance to piperacin looked like over the year. And again, it's now more than 10 years, but we do know the picture. Artemisinin was already fairly high prevalence at the time in the region. Piperacin goes up quite rapidly and pick up to reach pretty much took the almost complete prevalence in the region. This is an important picture because, as I was saying, gives you a snapshot in time of what is happening. Although we do know that there are other ways to get the same picture. For example, we've heard. Sorry, I'm coming down with a cold, so apologies if I'm coughing. There are other ways to get to similar conclusion, probably even more accurate, you might argue, by therapeutic efficacy studies, clinical studies in the region, and so on. So far, nothing new. But what this kind of analysis led us to do was actually looking under the hood. Let's open this parasite quite literally, in fact, and let's see what this increase in frequency really looks like. What is going on? Is it still the case of, as I was saying at the very beginning, in the very first slide, of these mutations coming up independently in different parts of, in this case, of the country, later on in the Greater Mekong Subregion, or there is something different there? To do that, we use what is called statistical chromosome painting. I'm not going to go in details of what that really means, but the bottom line is we're trying to analyze haplotypes, that is the part of the genome in and around the Calc13 locus, and later on also in and around the plasmapsin locus. And we assign each sample to what we call genetic type of resistance. What I mean by that is a group of samples that has the same resistance mutation, strong haplotypic similarities, that means the genome in that part around Calc looks pretty much the same. And when you take these two things together, that means that there's probably a recent shared, use the word lineage at the locus, as in there is relatedness between these parasites, at least as measured locally. To a first approximation, although it's not completely correct, you can imagine genetic types as different molecular origins or resistance. Now, remember that Plasmodium is a sexually recombining organism, so the analogy with COVID doesn't quite hold, but you could almost imagine this I'll try again. These epidemiological lineages, like the ones we see in COVID, like imagine those as, I don't know, the Delta and Omicron and so on, BA1, BA2, and so on and so forth. Again, don't read too much into the analogy. It biologically just doesn't work, but it is a good concept to keep in the back of your mind. So in total, we identified more than 30 types of artemisinin resistance and at least 3 different types of piperacin resistance. So in the figure on the left, I'm showing the haplotypes that surround the Kelch13 locus. Kelch13 is here in the middle. And this is in about 500 Western Cambodian samples, which are represented on the row, as in each row is a different sample. In orange and in pink, I've highlighted the samples that belong to the 2 most common types of artemisinin resistance. The most by far the most common one being what we end up calling KEL1, which are these parasites here, which, as you can see, they're very similar from a genetic standpoint. You could already see in the previous slide, but let me stress even more and take this point home. Not all types were created equal. Some were more common than others. One of these types, which again is a specific origin of Kelch13 mutation, which we call KEL1, and in fact a specific origin of plasmodium amplification, PLA1, rapidly became dominant and was the main lineage responsible for treatment failure in Western Cambodia during those years. So this plot on the left tries to unpack these dynamics in a bit more details. The thick black line is the same I was showing a few slides ago and depicts the overall trend of artemisinin resistance. Of course, in Western Cambodia between 2007 and 2012. You can distinguish 2 distinct waves underneath the line. The first one, when the KEL1 type in yellow became dominant. You can see it starts as a fairly lame type at the beginning. There's much more going on, but then rapidly picked up and becomes pretty much the dominant lineage. The second wave, which is here in yellow and red lines and yellow and red dashed lines, is when there's 2 types. The specific origin of artemisinin resistance I'm talking about, KEL1, and the specific origin of piperacin resistance, which I'm calling PLA1, come together and essentially wipe out completely the competition. Again, loose analogy with COVID, you can see KEL1, PLA1 as, for example, Delta, which took over, at least in UK, the COVID population before Omicron came around. But that's Western Cambodia. What about the rest of the region? Unfortunately, the spread of this type of resistance didn't really stop to Western Cambodia, but spread across the national borders into neighboring countries. And in some cases, and I'm showing the frequency of again, this KEL1, PLA1 type in 6 regions in the in 6 areas of Greater Mekong Subregion. You can see in some places like Northeast Thailand or North Cambodia, actually KEL1, PLA1 reached the frequency nearly 100%. That means KEL1, PLA1 had completely overtaken the local population, completely wiped out the local population. The reason why one of the reasons why I'm talking about KEL1, PLA1 here is to, again, credit to GenRe-Mekong project. Once you've done all the work in identifying these types, again, think about COVID. Once you've done the work in identifying what Omicron really means, now you can actually roll it out and being part of routine surveillance. For example, even with Amplicon, which is what Olivo and colleagues have been doing for a while. That means, again, that allows to have high resolution picture like the one I'm showing here, or the one even higher resolution that Olivo was showing at the very beginning of this forum. Now I'm talking about KEL1, PLA1 spreading. But I haven't really quantified that statement. So is it really like that? So if you take KEL1, PLA1 parasites from across the region, no matter where they are from, they all look more similar to each other than non-KEL1, PLA1 parasite. That is, you take KEL1, PLA1 from Vietnam and you take one from Laos, those KEL1, PLA1 parasite actually looks, you can still see at a genomic level, they're very, very similar to each other. They're very close genetically. Much more than, for example, if you were to take a KEL1, PLA1 parasite from, let's say, Laos or Vietnam, and compare it to another parasite, which is not KEL1, PLA1 from Laos or Vietnam. So basically, KEL1, PLA1 form one sort of family. I'll show in a second that this analogy doesn't quite work. There is more differentiation than that. But still, you can see this family of parasite being related and spreading in the region. Even a few years later, you can still see sort of ancestral signature of the original spread. I've used the analogy with COVID a few times now, which I think makes for good way to contextualize results. But I've also tried this is really, really not accurate. This is particularly evident when you look at genetic similarity within the KEL1, PLA1 group. As I was saying, regardless of the region, they are related. But actually, when you look within that group, when you look now genetically within the KEL1, PLA1 parasite, you start distinguishing what we call subgroups. You can almost imagine as in subfamilies, or I don't know what the correct terms is. Keep in mind, these are recombining organisms. So just want to stress out, this is not a pedigree. This is not a family tree. This is just a visualization and nothing more than that. What I want to point your attention to is this group of parasites, which are incredibly similar from a genetic standpoint, hardly have any difference. In fact, when you look at the entire genome, they are all clones of each other. I'm not making any claim of how they are related to each other across subgroups, but within each subgroups, you can see there is almost complete identity. What was particularly worrying is that each one of these subfamilies seems to carry additional mutation elsewhere in the genome, in particular in CRT. And independent clinical study led by Rob van der Pluim and Moro at the time shows that those mutation, those CRT mutation, again, survival curves on the right, do have a more negative outcome than parasites that don't carry those alleles. So the picture starts to get a bit more complex. And speaking of complex picture, I'm going to show an incredibly complex one. So please bear with me. You can imagine that the reason why this slide is half empty is because it's going to get incredibly busy in a few seconds. I'm going to take you step by step. I'm going to take one bit at a time. So what I'm going to show on the left is the CRT gene. As I was saying as I was saying a few seconds ago, mutation in this gene seems to, in addition of KEL1, PLA1 lineage, seems to give an even more negative treatment outcome. Gray dots are mutation actually happen to be a background, the CRT genetic background, which we know very well is actually the same genetic makeup for this CRT gene that we find in the parasite called DD2. This is a known lab strain. It's got a certain number of CRT mutation, in particular 7060, we know as chloroquine resistance, plus something else. Then I'm showing in the middle the proportion of samples in 4 different populations from Asian populations that carry the haplotype, and finally the number of samples in the new data release is coming out in a few weeks, 20,000 parasite samples from all around the world. Again, in that release, this is the number of parasites that carry the specific haplotype. Now, hold your breath. This gets very busy, but don't be scared. You can see that the first haplotype I was showing, the DD2 CRT haplotype, is by far the most common. Please note this is a log scale. So there are thousands of parasites in the PF7 release that carry that haplotype. But right after that, there is a number of haplotypes, and let's focus on, just for the sake of argument, on the first 4 or 5 here, which carry additional mutation on top of that background. So this is, if we look at the second row here, this is a KEL1 PLA1 parasite, which has got a DD2 haplotype in CRT, plus an additional mutation, in this case, T93S. Again, one of the mutations identified by Rob in the clinical study. The good news, if there is such a thing, is that if you look at the prevalence, geographic prevalence of this parasite that carry this very complex genetic architecture, seems to be constrained to what we call East part of Southeast Asia, that is Cambodia, Vietnam, Laos, and eastern part of Thailand. So that's good news. And by the way, all credit to Richard Pearson, Anastasia Hernandez-Koduchev, and Jacob Almagro Garcia for this analysis. I'm running out of time. I just want to leave you with one final finding, which I'm going to put in the category of good news, assuming there is such a thing. We were already seeing that switching to artesunate mefloquine at some point was beneficial, as in was working. I want to supplement that with additional finding. This one, again, is in vitro. And this is to stress, again, why we care about KEL1 PLA1. Well, one reason I've already said, we can track those KEL1 PLA1 parasites. We know they're DHA PPQ resistance, and they give us a good handle, a good hook to see how they're moving across the region. But the other reason is that as it turns out, they might actually have, and I'm using the conditional here, some genetic makeup that make them more sensitive to, for example, mefloquine, as I'm showing here. So we know that MDR1 copy numbers are associated with mefloquine resistance. That is, parasite with a single copy have a low mefloquine IC50. Parasites with multiple copies of the gene MDR1 are resistant to, or what canonically is considered resistance to mefloquine. But look at the KEL1 PLA1 parasites. They are so low on this plot. Means not only they're sensitive to mefloquine, but to use an analogy from Olivo, actually, you don't even need to use mefloquine. You can just show the package of the drug, and those parasites go. So again, these are unpublished results. We hope to wrap it up. And in reality, what's really important is what is happening in the field and whether these drugs are working or not. But again, taken together, that shows why this nomenclature kind of works, why it's useful from a surveillance point of view. Just want to leave you with take-home messages. I think it's very, very clear from everything we heard so far that the emergence of anti-malaria drug resistance is a matter of when and not if. That means we need to be prepared. And timely intervention are critical. By the time we've done all the clinical studies and so on, actually, KEL1 PLA1 had already spread in a number of countries. So, and this connects to what we're going to hear later in the day and tomorrow. We need ways to decipher the trajectory at the very early phases of resistance, as in as soon as possible, so that we can stay ahead of the game. And again, we're going to hear some great stories about the characterization of the outbreak in Laos. Just want to finish by thanking an insane amount of people that have been somehow responsible for this work in one way or another. I'm not even going to try and name anyone. Big thank you, but a special mention to the GenRe-Mekong project, as in this collaboration with control programs has been by far the main reason why I've been able to look in details at the dynamics of spread of resistance in the region. And thank you all for listening.
Okay, well, thank you very much for that wonderful presentation. It was quite a ride, I'd say. And I had forgotten I said that thing about the packages, but never mind.
One more question to the speaker. That's we are talking about the KEL1 and PLA1 that is so the high sensitivity. compared to the looking for the prediction of resistance. So what is the level of sensitivity we are talking about? And I don't know that you're talking about high sensitivity. So what does mean high sensitivity compared to the one that we already might only see the C580? Thank you.
Sorry, which speaker were you referring to?
I can take that, Oliver, if you want.
Oh, sorry. Yes, Roberto.
So we only have molecular data for that. And I mean, credit good to Parat Lim and Chanaki Amaratunga for collecting this data. In lab, these are ex vivo IC50 experiment. We see that you need about half of what we see about half of the IC50 that we'll see for normal sensitive parasite. That is, on average, you need 20 nanomolar, if I remember correctly, for sensitive mefloquine sensitive parasites. KEL1, PLA1 seems to require even less than that, in the order of 10 or so. But again, I want to stress these are in vitro study. What that translates to in terms of clinical practice, what that translates to in terms of public health, that's a big open question.